Imagine identical twins, Sarah and Emily, sharing the same twinkling eyes and infectious laugh. They share the same DNA, a blueprint for life, yet their life stories unfold in surprisingly different ways. Sarah thrives, embracing a healthy lifestyle, while Emily battles heart disease in her 50s. This begs the question, if their DNA is identical, why are their health outcomes so different? The answer lies in the fascinating world of epigenetics. Think of our DNA as a vast library filled with books containing the instructions for life. Each gene, a single instruction manual within those books, holds the potential to build and maintain our bodies. However, not all genes are actively expressed or switched on all the time. This is where epigenetics comes into play. Epigenetics acts like a series of bookmarks, highlighting which genes should be read and acted upon. These bookmarks don't change the underlying DNA sequence, but they influence how our genes are interpreted and expressed. Our environment and lifestyle choices play a crucial role in placing these epigenetic marks, shaping our health and well-being. Imagine a bustling city with traffic lights directing the flow of vehicles. Similarly, epigenetic marks act like traffic lights regulating the activity of our genes. These marks can either activate or green light or silence or red light specific genes, influencing the production of proteins essential for various bodily functions. Just as traffic lights ensure order and prevent chaos, epigenetic regulation ensures that our genes are expressed at the right time and in the right amounts. One well-studied epigenetic mark is DNA methylation. Think of it as attaching a sticky note to a page in our DNA book. This sticky note, a methyl group, doesn't erase the underlying genetic text but signals the cell to either read or ignore that particular section. Another crucial epigenetic player is histone modification. Histones are like spools around which our DNA is wound. Modifications to these spools can either loosen or tighten the DNA wrapping. Loosely wound DNA is easily accessible for gene expression, while tightly wound DNA remains hidden and inactive. These fascinating mechanisms highlight how epigenetics fine-tunes gene expression without altering the DNA sequence itself. Our lifestyle choices like diet and exercise play a significant role in shaping our epigenetic profile. Imagine a group of construction workers following blueprints to build a house. Similarly, our cells rely on genetic instructions to function correctly. Now envision these workers receiving additional instructions, modifying their building plans. Lifestyle factors act like these modifications, influencing how our cells interpret and utilize genetic information. For example, a diet rich in fruits and vegetables provides our cells with essential nutrients that can influence DNA methylation patterns, potentially activating beneficial genes and silencing harmful ones. Conversely, a diet high in processed foods and unhealthy fats can lead to detrimental epigenetic changes, increasing the risk of chronic diseases. Physical activity also plays a crucial role in shaping our epigenome. Exercise triggers the release of signaling molecules that can modify histone proteins, influencing gene expression patterns related to metabolism, inflammation, and even mood. Engaging in regular physical activity is like fine-tuning a complex orchestra, ensuring that our genes are playing in harmony. For years, scientists believed that traits acquired during our lifetime couldn't be passed down to future generations. However, emerging evidence suggests that epigenetic changes in some cases can be inherited, adding another layer of complexity to our understanding of heredity. This means that our lifestyle choices today could potentially impact the health and well-being of our children and grandchildren. Imagine a chain of dominoes, each representing a generation. Just as a single push can set off a chain reaction, certain environmental exposures or lifestyle choices can create epigenetic marks that are passed down through generations. For instance, studies have shown that children and grandchildren of individuals who experienced famine during childhood may have an increased risk of developing metabolic disorders later in life. This intriguing phenomenon suggests that our experiences can leave a lasting legacy on our descendants' epigenomes. While more research is needed to fully understand the extent and mechanisms of epigenetic inheritance, these findings have profound implications for public health and disease prevention.